Okay, so welcome back everybody to this, our first de-risking session. The first panel is going to be chaired by David Gann. He's the Vice President at Imperial College, and for our sins we both sit on one of Boris's Science and Technology working groups, so we have the pleasure of seeing each other every 3 months or so to talk with the City and the GLA and London and partners about the direction of travel in London, MedCity and the wider UK sector. So that's how David and I know each other. So without further ado, we've still got a few people coming in, but I'm going to hand immediately over to David, who will introduce his panel and set the scene for the session. Thank you, David.
Thank you, Harriet. Good morning, everyone. I hope you enjoyed the plenary session this morning with the keynote speakers as much as I did. I found it really, really interesting and stimulating. Our session now is focusing on de-risking research and trying to articulate collaborative models. And, well, there's a big topic for us. And I thought just to start off with, I'd say a few words. This image here is part of a new campus that we're building at White City in London. It's a £3 billion, 25-acre development that will take us some years into the future to complete. If you pass up and down the A40 today, you will see some rather large buildings coming out of the ground, one of which will be completed by this time next year and ready for occupancy. And it's this building here you can see on the left. And this building is being designed for collaborative research. So I thought I'd show an image of it, and I hope that some of you at some stage will collaborate in it with various other institutions and players. And my role at Imperial is in part to try and change the way in which we work together, knowing that no university or institution or company can solve the great challenges that we have before us in any of our main areas, whether it's environment or health and well-being, as we're looking at today, and that we need spaces to work together and combine expertise. So there's a physical manifestation. I hope we're also going to hear about some of the organisational and social processes that bring teams together. And I just give you one other image here, and I use this when we're looking to bring teams together. Some of you will know Chris Tumazu, I suspect, one of our biomedical engineers. And he's sitting in what we sometimes call the sweet spot. And if you think along the horizontal axis, the timescales of developing technology, the innovation process, you move from what the know-how we have today through new technology through to real breakthroughs that are in the realms of science and discovery. And on the vertical axis, we have today's markets. We have newer markets in adjacent spaces, and then we have unknown markets. Now, this is a fairly generic diagram, but of course, in drug discovery, we have targets, and we try and solve problems for desperate need of people with awful illnesses, as we heard this morning. But sometimes scientific breakthroughs throw opportunities and we don't know where we're going to use them next. Viagra is a good example. Chris Tumazu has found ways of collaborating somewhere at the upper right-hand side of this diagram, where most of us in the academic realm wish to inhabit, and I suggest we stay there because keeping a focus on the fundamental science and ideas is a very good thing to do, in my view, and I don't mind putting that out as a contentious statement from the chair. A very good thing for universities to do, because collaborating with industry is about bringing collateral together and having complementary assets, we call them. And those assets, we have to have strong science and fundamentals. And Chris, just to wrap up on that example, has developed DNA electronics, which is a lab on a chip, which has wonderful sensing fields, 350,000 sensors, and it will do gene sequencing. And to do that, he had to bring about collaboration across the academic disciplines first, in the top right-hand corner. No mean feat for any of you who've been trying to break the mould of the 20th century discipline base of our universities. And then collaborate through into the health sector to get tests done. And his lab on a chip will do DNA snips of microbes in microbial infection, hospital-acquired infections. And his aim is to reduce the time of diagnosing what microbe is giving you sepsis from whatever it takes, 36 hours if it goes out to a path lab and comes back, to just an hour there and then in the hospital, so you can blast that microbe with the right antibiotics. And there's a link there directly then to how we understand drugs, because you've got a sensing device in the theatre that you can use. And that takes a lot of collaboration to get out into practice. So I leave you with that just as a framing, and take it or leave it. Frankly, I'm chairing the rest of the session, and I just wanted to stimulate some thoughts about collaboration and say there's a sweet spot somewhere there, and let's try and understand it, and let's hear then from our first speakers. We've got a great lineup here, and I'll read out the names in a slightly different order, because Steve McMahon from King's College London is going to have to leave us, and so we've organised ourselves to have his talk for 10 minutes, and then a bit of a Q&A from everybody, and then we'll roll back through with Sir Philip Cohen from the University of Dundee, followed by Stuart McElroy from European Screening Centre Newhouse, and then John Moore from Horizon Discovery. And I've asked our speakers to keep to 10 minutes or thereabouts. I hope we have a fun conversation with you all as we go through the rest of the morning. So Steve, over to you. Thank you.
Well, thank you. So I'm an academic from King's College, and I've been involved in translational research for quite a long time, and I've been asked to talk about some of my experiences at trying to do this research in a collaborative way. And if I could have the first slide, please. So I think collaboration does have a bad rap sometimes. So the whole idea of kind of co-production, and it's supposed to bring out the worst in everybody. And I'd like to say that actually that's not my experience. Mine is the exact opposite of that, and that's that collaboration has a fantastic opportunity to add value. And I'd like to I'll give you a few examples of how I've seen that in operation. So I've been involved in several collaborative efforts over the years, which I list here. They're all based on pain research, and pain research, to pick up at the earlier discussion, is an interesting area of huge unmet clinical need. Because people don't die of pain, the regulators set a very high bar, so it's very difficult to bring new treatments to market, and frankly, we've been pretty unsuccessful until the last few years in doing translational medicine, although the thing is improving now. I've been involved in several efforts. They all have many similar features. I would say as they get bigger, they obviously get more difficult to manage, and that is something of a problem. But I thought I'd give examples from this effort. This is one that we started about 5 years ago. We were one of the first people funded under the EU Innovative Medicines Initiative, with a grant from the EU matched by quite a lot of money from private companies, FBA companies. So it's quite a large activity, and the goal of this whole activity was to increase our understanding of pain mechanisms and improve the treatment of chronic pain patients. So I'll tell you a little bit about who we were. We're quite a number of groups. So these are the academic partners. I think we were lucky that we weren't just random people from around Europe in this case. We were already grouped into sub-consortia who worked together. So the UK group were all part of the London Pain Consortium that had been working together for 10 years. And similarly, there was a German network and a Danish network who also had quite a bit of experience of working together. The FPA partners were, they've changed a little bit over the years, but they were all the usual suspects, and they're all drug companies, most of the drug companies in Europe, which have or had an interest in treating, developing new therapies for pain. So the organisation we had is kind of illustrated in this slide. And I think this is the first point, is that, I mean, obviously the drug companies are competitive, and that is a bad thing for collaboration. So we needed to find a mechanism, and that was the whole point of the EU IMI scheme, to find a mechanism to allow FPA partners, pharmaceutical companies, to share their expertise and knowledge, so to find some pre-competitive space. And so that's what we did with the FPA companies. For the academics, again, I think the last thing you want is a bunch of academics competing with each other. So the value here is to get people who have complementary knowledge, which allow synergies and value to be added. So the details are not so important, but our work package, our work scheme involved a lot of preclinical work in animals, and it involved a whole number of studies in human volunteers and in patient groups. And so it was organised roughly into different work packages, and each participant typically participated in 2 or 3 of these work packages. So the first point is that in any one box, which was really the unit of activity, there were a team of maybe 8 or 10 people who met regularly to tackle the problems within that box. But the whole point of these boxes is that they're interconnected. But what was happening in this box was meant to inform what was happening in the next box. And whilst that sounds very easy on paper to say, it really did work that way. So the first point is that what you get from working collaboratively is synergy, that you get everyone's expertise that can add together to more than the sum of the parts. And I thought I'd just give one simple example of that from our own work. So one of the questions that this consortium was interested in was what causes pain. So we know that many disease states are associated with pathologies in peripheral tissues, and the evidence is that the peripheral tissue is making some chemical mediator which is driving pain behaviour. And actually, I mean, our ignorance is that we don't know what most of those mediators are. And certainly the drugs that we have to treat existing mediators have a relatively limited efficacy and quite a lot of side effects. So there's a real need to find new mediators. So as a team, we've tackled that. It's actually an ongoing process, but I can give you one example of how these synergies worked. So it turned out at the beginning of this grant, I had been working on a model of UVB inflammation in animals, so that we could create this model and we could characterize it, and we were using it to study pain mechanisms. Very quickly, when we started this collaboration, we teamed up with some of the German workers to implement this model in human subjects. In 5 minutes, yeah? In human subjects. And then we additionally used the emerging omics that was present in many of the FPA partners. They had taken on that technology very early. So we applied omics to samples taken from these human volunteers, and simultaneously we took them from some chronic pain patients. And then we used our bioinformatics capacity to try and identify from these omics candidate molecules that might serve as pain mediators. And then we took those mediators and we went back and tested those out in preclinical models. So it's a kind of reverse translation which led us to novel mediators that we're now studying clinically. So in fact, this effort was published a couple of years ago, and we are now trying to pursue trying to develop an antagonist for a new target, CXCL5, a chemokine, which we implicated in the cause of generating some of this pain. So I know the details will all be a bit blurry there, but the point is that by interacting, the synergy, the different expertise that you can bring to the table really makes things possible that would not be possible. So that's one example, and I thought was the obvious point of collaboration. That example was not unique. Around this diagram, we've listed all of the partners, and all I've tried to do, I know you weren't going to see the details, that's the point really, there are lots and lots of interactions that were present. This was after 4 years of existence of our IMI. And you see that we facilitated those interactions by a lot of exchange visits between partner groups. So I think that synergy is the obvious point. A less obvious point is this one. Which again is an ongoing effort from the IMI. And this is, I think it's a brilliant example of how to do pre-competitive research. Many of you will know that drug companies are notoriously secretive about their data, and they think they're giving things away if they share that. But actually, there are advantages to sharing. So look at this. One of the things that we got the drug companies to agree to do was to put into a pot lots of data from clinical trials that had not been fully published so that we could do a meta-analysis on the combination of those data sets. And that's going on now, and some of it's taken place, but clearly you have a lot more power in the cumulative data sets than you do in the individual data sets. So one of the advantages here for the pharmaceutical industry is that by giving a little, they get a lot more back. And I think that's, again, it's sort of a groundbreaking development that is only possible in the context of a sort of trusting environment. And the third and final example I'll finish with, I think, addresses something which is truly poisoning clinical research and preclinical research, and that is the lack of reproducibility of what's published in the journals. So if you haven't seen this article, I would recommend it to all of you. It's a disclosure by drug companies about how difficult it is to replicate particularly high-profile publications in the academic literature. So one way to crack this problem is to work collaboratively. So my third example, which was adopted, not by me actually, by other members of this IMI, was to set up a kind of a preclinical multi-center trial of a new strategy. So one of the problems we have in the pain field is that it's difficult to know whether an animal is in pain. We can't measure spontaneous pain in animals very easily. One method that's been proposed is to use burrowing activity. So it's a new development. It doesn't really matter what the development was. The point is, how do we know whether we can trust the results of a single study? So what this consortium did, it was quite a tedious event actually. It got 8 different laboratories around the world to sign up to a common protocol. And the protocol was quite long. It involved lots of training, lots and lots and lots of teleconferences, but it ended up with a common protocol that was implemented in all of these groups to see whether or not this burrowing was fit for purpose. And the global result is shown here, and what we're plotting, the red and the blue lines are controls, and they're flat. The black line shows a nice response. We can see a change in behavior, burrowing behavior, which we think is consistent with an indication that this method picks up the spontaneous amount of pain that animals are feeling. So that's very nice. So the first thing we did was we validated a new approach. But the really interesting thing happens if you look what happens at each of the 8 centers. So I think this is just fascinating. Right? All these people are following exactly the same protocol. They've all been trained to do exactly the same thing. There were some protocol violations because some people couldn't get the right gravel. I mean, there was necessarily some variation. But look at the result. Clearly, 2 centers failed to replicate any effect in this model using an identical protocol. All the others did replicate the main effect, but to different degrees. So I think this preclinical approach, which is only possible in these consortia, is very powerful. So one, it tells us that this method is effective. It also tells us that it's likely to be sensitive to a number of very mild, unknown variables that we can't capture. So obviously this will be a tedious process to follow for the whole of preclinical development, but I suspect in order to overcome this problem of lack of replicability, we will need team efforts along these bases which allow us to get to the truth in a single step, or getting closer to the truth. We never get to the truth. Okay, my time is almost up, so what do I think about collaborations? I think they're good for a number of things. So they're very good in making you competitive. If you get a bunch of people together who are all highly regarded, are very proficient in their field, the team they make is very, very powerful. So we have received a fantastic support from a number of organisations, particularly the Wellcome Trust actually, partly because we can make a very, very competitive case for what we want to do. We're credible. When we do things, people listen because, again, it's the voice of a large number of experts in a single field. I think the most obvious value of collaboration is that you add value to individual activities, and I think that's of itself would be an important step. I also think, and as I hope you've given you some examples, that you can innovate by simply putting together different bodies of knowledge. You can do things that you couldn't have done individually, whatever your inclination. So I think our meta-analysis is a good example where we get more out than we put in. And of course, all of this comes at a little bit of cost, and the cost is a bureaucratic one, and that one has to be prepared to spend time developing these common aims. So it does mean, I think it's bad for some scientists who want to be superheroes, me. If you give up a bit, though, everyone has to give up some of their time for this team effort. But my experience has been that the results of that team effort more than outweigh what you individually put into it. And if anyone's interested in more details about this particular private-public consortium, they're available at these sites. So thank you very much.
Thank you.